Good evening, everybody. Welcome to our third of 7 webinar presentations of the UC Santa Barbara Natural Reserve System. We're so glad to have you join us this evening, and boy, are you going to be in for a treat. Tonight's lecture will be focused on research that was carried out at UCSB's Coal Oil Point Reserve. Coal Oil Point Reserve is one of 7 reserves that UCSB manages that's part of the UC Natural Reserve System. What is the UC Natural Reserve System? Well, there's a series of 41 sites across the state of California, and they are protected lands explicitly for the purposes of research, university level education, and public service. Each UC campus manages a handful, and we here at UC Santa Barbara manage 7 of them. This webinar series gives you a tour of all 7. And last week we heard from Dr. Adam Green from Santa Barbara City College, presenting on behalf of the Carpinteria Salt Marsh Reserve. And as I mentioned today, we're moving just a little bit west from there over to Coal Point Reserve. Just a few housekeeping items before we get going tonight. We will have tonight's format will be that we have a 15-minute presentation by Dr. Cris Sandoval, who's the director of Coal Oil Point Reserve. She will introduce the reserve and also introduce our speaker tonight, who is Dr. Sarah Weinstein from University of Utah. After Sarah presents for 30 minutes, then we will have a 10-minute Q&A, and that is your chance, the audience, to ask your questions of either our speaker, Dr. Weinstein, or our reserve director from Coal Oil Point, Dr. Sandoval. Please use the Q&A function, which you can see at the bottom right hand of your screen. There's a little button that says Q&A, and that's how you'll type in your questions that you can ask to our speakers. I will moderate the Q&A session, and we usually get so many questions that I won't be able to get to all of them tonight, but we'll make sure to send them to our speakers in case you're all trying to get responses to your good questions. Just another note, this webinar is going to be recorded. So if you didn't catch the whole thing tonight, or if you want to share this with others, it will be posted on the UCSB Natural Reserve System YouTube page tomorrow. That's where all of our other lectures are also posted as well. So you can catch up on everything that you've missed already. With that, I'm going to turn it over to you, Cris, and why don't you get us going?
Good evening, everybody. I hope you had a good dinner and you have a glass of wine in your hands, and hope you enjoy the next hour with us. Today, I'm going to give you a really brief overview of Coal Point Reserve with the intention to get you here to visit us and see what we're doing. And then we'll hear about this wonderful research by Dr. Weinstein. But first, I'd like to start telling you a little bit about how I got here. I know we have quite a few students in the audience, and you may be by now having some anxiety and wondering what I'm going to do next and how I get a job like yours. So I'll tell you really quick what was my trick. So I came from Brazil 30 years ago to do my PhD at UCSB. And I meant to go back to Brazil, but I met a guy and I met a place called Coal Point Reserve. And I'm still here. So at Coal Point, I started as a volunteer while I was doing a postdoc, living in a trailer, but getting free rent in a nice location. It was not my original plan, but I started really liking it. I soon realized that the reserve was the place that I could fulfill a personal philosophy, which is that research and education are means to save the world from environmental destruction. And conservation has always been a passion of mine since I was very little. And when I did my master's in Brazil, I realized that research could help. And that was when the 2 and 2 clicked together. So I'm still at Coal Point Reserve because it was the right place to fulfill this vision. So the lessons that I learned were, a tip for you guys, young students, is to be really flexible about unusual opportunities that may come up to you, but also to stay focused on options that match your passions. Don't ever let go of your passion. So now I'm going to share my screen. Oh, you already have my screen there. Yeah, great. So Coral Point Reserve is a coastal reserve. It's number 30 on the map there. And it's 4 miles from UCSB campus. And last week, you heard a talk about Carpinteria Salt Marsh Reserve, which is also a coastal reserve. And you might wonder, why do we need two coastal reserves? And well, they couldn't be more differently. So Carpinteria Salt Marsh is fully tidal, means there's a tidal system throughout the year. It's dominated by a salt marsh, has no dune system, is dominated by a salt marsh snail that really affects the whole ecosystem. And there's no endangered tidewater gobies there. Coral Point Reserve is seasonally tidal, meaning it's only tidal during the winter rains. And it's dominated by a mud flat with a very extensive dune system behind it. We don't have the snails, but we have tidewater gobies. This kind of reserves that are close to each other but so different allows researchers to compare and understand why nature is the way it is, and ecology. They can do ecology and evolution, all sorts of comparative studies with reserves like this. Let me just figure out why this is not moving. Nope. Ah, got it. And here's a slide of the reserve and the team that we work together to make this beautiful reserve even better every day. Jessica Newson is the conservation coordinator, and Keith Callahan is the steward and botanist for the reserve. We also have Tony Nelson, who does our IT work. As you can see on this slide, Coral Point is in the middle of an urban area. So a lot of what we do has to do with dealing with that relationship between being a protected reserve and having the pressures of urbanization nearby. But within this really small reserve so close to campus, we have a lot of different habitats, very special ones. Coastal scrub, the Devers Slough, vernal pools, coastal bluff, salt flat, dune pond, and my favorite habitat is the sand dunes. As you can see, it's just that absolutely gorgeous habitat. And also there's so little of it. Think about in the entire world, there's so few dunes out there, and most of them are impacted by invasive species. What all these habitats do is provide an incredible opportunity for researchers and students from campus to come here in a 10-minute bike ride and do their research and their explorations and learn and practice field methods. For one of our probably most well-known program is the Snowy Plover program. And I'm so excited to tell you that we are celebrating 20 years this year. I can't believe it's been 20 years since we found our first Snowy Plover chick at Coral Point Reserve. And during this 20 years, we provided over 230 stipends for undergraduate students at UCSB to join the program. We had about 50,000 hours of docents on the beach. These are people from the community, they are students, faculty. They just like to volunteer to be there and help inform people about the program and this wonderful species. And as a result, we got 1,400 snowy plover chicks hatched so far. This is just so incredible. It's not always easy to babysit an endangered species. Just this last year, we had to resort to two creative solutions. So we're always having to outsmart predators here. We had to create this cage that is double walled to prevent predation from gulls and from crows. You can see the little plover sitting in the middle of the cage, sitting on this egg while the gull is lurking around, see if he can get the eggs, but he can't put this cage around. And then on the right, you see Jessica, and what she's doing is she's swapping the real eggs with wood eggs. And we're going to keep the real eggs in an incubator. And the plover does not know the difference and sit on the wood eggs for 30 days. And as soon as the egg in the incubator is starting to show signs that it's going to hatch, and they actually, you can hear it, you put the egg on your ear and they go beep, 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 beep. And then you know it's time to rush to the beach and do the swap again. And that was a great way to trick the skunks because the skunks figured out a way to get through that cage. But with the wood egg, they threw a few and they gave up on it. Another great program we have is restoration. And this is Kip here demonstrating to students how do you take the plant out of the tube, and he teaches the students about native and exotic species and how do you plant them and which species do we plant in which habitat. And it's just a wonderful program too that has a lot of people involved. We have restoration days. In fact, if you guys want to form a group and you want to plan things and feel good about what you do, just let us know and we can create a work day for you to join us here. And some people really like to pull weeds. And here's a group of students really happy after a long day doing restoration. I want to tell you a little bit about what kind of research goes on at Coral Point Reserve. And there's a lot of different types of research. And this is Samantha. She did a senior thesis trying to understand how mammals move around Coral Point Reserve. She wanted to know, do they use the trails that people use, or do they prefer to be hiding on the bushes and going in difficult places? The answer is they prefer the trails, but they do it at night. And this is another group of students who, after the 2000 oil spill, they wanted to understand if this spill affected the intertidal habitat. We have lots of faculty also involved with Coral Point. This is Professor Hank Peacher, who has been painting Coral Point Reserve for many decades. And those paintings provide historical documentation about how the changes that happen at Coral Point Reserve. This is a group of researchers monitoring their intertidal. They wanted to know how the marine plankton settles in the intertidal and recruits into mussels or chitons or things like that. And I don't know if you know, but Coral Point Reserve on the Davros side has an amazing population of tiger sharks, sometimes leopard sharks, sometimes 1,000 individuals. And these researchers are actually taking the contents of the guts of the shark without harming them and putting the shark back in the water. And they can study what the sharks are eating because there's very little known about them, despite the fact that they are so abundant here. And here we have Dr. Jenny Dugan, who is an expert on beach ecology, sandy beach ecology, and she's been studying the community of the sandy beach and many different beaches throughout the Santa Barbara County. And then we have another research by Dr. John McLaughlin, who is studying the food web of particularly the Coral Point Reserve. But this food web is really special because it involves parasites as well. So not just they look at every organism that you can see with your naked eye, but then they also look inside of those organisms and see what parasites are in there. And we have here Dr. Cris and Dr. Kevin Lafferty. They're very interested in looking at eDNA, and they're trying to use that technique to detect species that are difficult to find. For example, we have tidewater goby, but sometimes they're in such low numbers that we can't catch them with a net. So by collecting samples of water, there might be pieces of eDNA that came from scales or the body of the fish, and they can detect that in a water sample. And this is Jessica again. we wanted to know if could we attract the California leasterns, which is another endangered species, to come and nest at Coral Point Reserve by using decoys and playback. So I hope that this gives you a good flavor of Coral Point Reserve, and I really hope that you come and you visit us. You can sign up for a guided tour, which we have first Saturday of each month, and can visit our nature center as well. And if you can't do that, you can also come and you can do a self-guided tour. Just download the podcast from our website and enjoy the 4-mile ride around the slough. And now it's my pleasure to introduce you to our speaker, Dr. Sarah Weinstein. And I'm going to read this because her history is too good to not say it right. So Dr. Sarah Weinstein grew up in Santa Barbara and went to UC Berkeley as an undergraduate where she studied diseases of salamanders. She discovered that fungus that is infecting frogs also infects salamanders. Ugh. She then came to UCSB to do her PhD with Dr. Armand Cris and Dr. Kevin Lafferty, where she studied the Raccoon Roundworm, and she has published several well-cited papers. Dr. Sarah Weinstein is now a postdoctoral fellow at the University of Utah, where she studied bacterial communities in rodents, including those found in giant poisonous African rats. Welcome.
Right. Let's see. Is that correct? The correct screen up there?
Yep.
Excellent. All right, thank you so much for having me. I'm really excited to talk about research that I mostly did at UCSB on studying raccoon roundworm, parasite avoidance, and the landscape of disgust. And I guess I'll start a little bit with a background about myself. As Cris said, I'm originally from Santa Barbara. I'm actually a proud graduate of Santa Barbara High School and one of the early graduating classes of VADA, the Visual Arts and Design Academy. I went to UC Berkeley for undergrad, where I planned to double major in chemistry and the practice of art, and ended up finishing with a degree in biology and ultimate frisbee. After undergrad, I actually worked as a field technician for a couple of years for a variety of agencies, including the National Park Service and USGS, working in truly horrible locations like Hawaii and Yosemite National Park. After a couple of years of that, I moved back to Santa Barbara to do my PhD at UCSB, where I studied parasites, mostly in raccoons, but also got the opportunity to travel to a number of other incredible locations to study worms, which I may be biased, but I think are pretty awesome. Now I'm a postdoctoral researcher at the University of Utah, after having spent about a year living in Kenya studying these crazy giant poisonous rats. I won't be talking about them tonight, but I'm happy to answer questions about that research later. And so before I actually get into the research, I actually want to start with the acknowledgements. This work would not have been possible without a huge team of people, most of them undergrads at UC Santa Barbara, as well as my lab mates and the UCSB Ecological Parasitology Lab and Coral Point Reserve. So we'll start with just a little bit of a background about parasites. When we think of an ecosystem, what species come to mind? What would you populate this landscape with? I think many of us, it's really easy to recognize that, the large animals, big things like predators, and think of those as natural parts of healthy ecosystems. But there's also a diverse array of parasites in these landscapes. And these infect pretty much every free living species that's out there. And there's a whole wide array of different transmission modes of things moving between the environment and animals, directly between animals, via trophic transmission when something eats something else. And so we've got all these really complicated life cycles that are oftentimes somewhat hidden from us. But these parasites are part of healthy ecosystems. And in many ways, they're actually very similar to predators. in the sense that there can be substantial parasite biomass in these systems. And if you were to pile up all the parasites, there'd actually be quite a bit of them out there, even though you can't necessarily see them. They have potential to regulate animal populations in the sense that, similar to how we think of predators controlling their prey, parasites can do something similar. And they're really a critical component of biodiversity. You think about every animal out there, wild animals, they all have parasites. And you see in the extinction of some charismatic larger animal, you're probably also losing, I would say, a charismatic population of small worms as well. And so something to think about, thinking about, loss of species out there. And that said, there are still quite a few parasites out there that maybe we don't want to encounter. Parasites that can infect humans, things we term zoonotic disease, and things that spill over from animal populations into our own populations. And we're all, I think, far more aware of these now than maybe we've ever been before. And this includes a variety of viral agents, but also things like worms, for example, Raccoon Roundworm, which is going to be the star of today's show. And while this is a relatively small fraction of the overall parasite diversity, these organisms can have substantial health consequences for us. And so there's a lot of interest in trying to figure out how to understand these diseases and how to potentially prevent them. And ultimately, understanding the disease dynamics of these parasites really requires understanding what the parasites are doing in wildlife. And because these parasites often have very complex life cycles, this sort of complicates these predictions and understanding how to control them. And so when we think about this, it's not just about one host, it's really about a whole community of animals. And so we need to think about these disease dynamics in a community context in terms of infection in the definitive host where the parasite matures. And also need to think about infection in other wildlife that, maybe the parasite doesn't mature there, but it can spread out, and you might find infective stages out in other animals as well. And then, of course, spill over to humans. And so that's really what we're going to be talking about today in the context of Raccoon Roundworm. And so to do that, we're going to first start about talking a bit about worms and raccoons, and then move on to worms and rodents. We're going to talk a bit about worms and us, and then think about how we and other animals can avoid these parasites, and finish off thinking more broadly about parasite avoidance and the landscape of disgust. And before I dive into the actual research, I just wanted to define a term I'm going to be using quite a bit today, and that is latrine, which is essentially the same thing in a raccoon context as it is in a human context. It's a communal defecation site. Raccoons make these. You may even see them on your property around. They're large piles of raccoon feces. And for raccoons, adult raccoons are the definitive host for this parasite. And so when I say definitive host, I mean the host where the worm reaches its adult stage. And as an adult, these worms produce eggs, which pass out into the environment, piling up at raccoon latrines, these communal defecation sites, which then sort of become hot spots of infection risk because they're piles of raccoon feces and also piles of parasite eggs. And these eggs can be picked up by other raccoons, particularly juvenile raccoons, which are more susceptible to infection. So we see transmission through the environment. And these worms are pretty long-lived, so a baby raccoon will grow up to be a teenage raccoon and then an adult raccoon that's still full of worms. So you get infection cycling that way. However, these eggs also can infect other species, things like rodents, which serve as an intermediate host in this life cycle. And when I say intermediate host, it's a host where the parasite doesn't reach its adult stage, but sort of serves as a go-between between this egg stage and then the adult raccoon. So when these mice that have larval worms in it are eaten by a raccoon, a process we call trophic transmission, the worm then is released from the mouse tissue and then completes its life cycle growing into an adult worm in the raccoon where it can produce more eggs, and the whole cycle completes itself. And this may seem fairly complicated, but really the important take-home message from this is that disease differs between hosts. So in raccoons, They're the definitive host. We have adult worms in the gut. These worms produce eggs. In raccoons, there's very little pathology. The raccoons are generally okay with this infection. In everything else, which is small mammals, birds, and humans, we fit into this life cycle as intermediate hosts, which means they're immature worms. They don't go to the gut. They stay in tissues. Parasites are only larval stages, so they're actually quite small, like about 1 millimeter, so really tiny. You essentially need a microscope to see them well. But because they stay in tissue and they have a tendency to head towards tissue, like brain tissue or the eyes, they can cause very high pathology. So typically associated with high pathology, but not always, depending on the host. And so keeping those distinctions in mind, ultimately understanding risk requires knowing about this parasite sort of in wildlife and in its natural system. And so to start, we need to know a little bit about the raccoons. For example, how many infected raccoons are out there? How many worms per raccoon? And how many eggs per worm? Because it's eggs that are kind of our unit of risk for us. And so to address these questions, I sampled a lot of raccoons for Raccoon Roundworm. Working in Santa Barbara County, I was able to dissect about 200 raccoons, pretty much all acquired from various pest removal services in town, as well as quite a few roadkill. And from these raccoons, I was able to get a couple of pieces of key information. So use the raccoon skulls to actually age them based off the skull sutures. Collected fecal samples, which allowed me to quantify the number of eggs that are being passed out into the environment, and then measured all the parasites in this whole parasite community within the raccoons. And ultimately, we see is that in Santa Barbara County, most raccoons have raccoon roundworm. In fact, 90% of them are infected. And what you're seeing on this plot up here on the x-axis is the worms per raccoon, and there we go, worms per raccoon. On the y-axis is the frequency, how often we see different numbers. And the majority of raccoons out there have very few worms, between 0 and 10. However, a small fraction of the population is heavily infected. There's a small number of raccoons out there that have a lot of worms. And it turns out that's generally the younger raccoons. So the juvenile raccoons, here we've got raccoon age plotted on the x-axis and the number of worms per host on the y. What we see is that juvenile animals have a lot heavier infection loads than adults. And this matters because some control efforts may actually end up shifting the age distribution of these populations. And if that happens and you end up with more juveniles, you might actually increase your risk of infection. And so we see that the number of worms varies between individuals. And then we take that one step further. We see that an animal that has more worms is also releasing more eggs, which makes sense because every female worm is producing eggs. And so I'm going to summarize that more worms equal more eggs, which ultimately equals more risk. And we know these infected raccoons are releasing huge numbers of eggs out into the environment, which then sort of begs the question of whether we see this parasite spilling over into other species, for example, rodents. And so to move on to this next question, are we seeing worms in rodents? So our next sort of understanding risk, linking up this life cycle, you can ask, do rodents contribute to this transmission? In particular, which rodents are contributing? Is it equal between native and invasive species? And even more important, if these eggs are just being picked up by rodents, but the rodents never get eaten by a raccoon, they'd actually be a dead end. So are rodents actually eaten by raccoons? And to do this work, this was all done at Cola Point Reserve, which many people think of more as a beach site, but actually has some incredible upland habitat as well. And so, working in this reserve that's got a range of different habitats and is also covered in raccoon latrines. Sorry, Cris. If you look at this map here, all those little white dots represent a raccoon latrine at the time we were doing this study. So this was about 2015. Most of them aren't there right now, which is sort of an interesting other direction I'd be happy to talk about later. But so we know there's a lot of raccoons out there, a lot of potential for exposure. And so we went out and we started trapping the mice and other rodents to survey for raccoon roundworm infection. And we see John Francis there holding a Sherman trap, which is the easiest way to catch a small rodent. There are these live traps that kind of boxes, and the mice go in, and then you can pull them out and they're totally fine. And yeah, beauty of live traps. For rats, we often use Tomahawk Traps, which are sort of the same idea. They go in, the animal's fine, it can be released later. These are kind of your typical rodent trapping approaches. And so we trapped rodents. There's 3 main species that you see at Colo Point. Rhythrodonomys megalotus, which is the harvest mouse, Paramiscus maniculatus, which is the deer mouse, and Rattus rattus, which is the rat and probably the rodent that's most likely to be found in your attic. The other 2, less likely to be in your house, although depending on where you live, you might encounter a Paramiscus. And so looking at the rodents and their parasite populations, noting that Rhythrodonomys and Paramiscus are both native species, whereas Rattus is not. It was introduced over 100 years ago, and they're abundant, particularly in coastal areas. And if we look at the parasite loads in these different species, so now we've got a plot where we have worms per mouse on the bottom here, and the prevalence, so the percent of the population that's infected on the Y. We look over at Rhythrodonomys, these harvest mice. We see very few worms per mouse, and very few mice are infected. Moving over to Paramiscus maniculatus, our other native species, we see that they're certainly infected. In fact, about 40% of the population has at least 1 worm, and on average is about 7 worms per mouse. I'm going to note that this is actually a log scale here. So moving over to Rattus, we suddenly see that the rats are incredibly heavily infected, with an average of nearly 1,000 worms per rat, and the vast majority of the rats out there having at least 1 parasite. And to give you an idea of what that looks like in one of these rats, if you were to look at the lung of one of these rats, you notice there's all these little spots there. Each one of those is a granuloma, which is sort of the host inflammation response to the parasite. And each one of those granulomas contains a larval raccoon roundworm. So these rats are incredibly heavily infected, which is really interesting because these are not a native species in this system, although raccoon roundworm, the parasite, certainly is. And so there's a potential that these invasive rats might actually be altering transmission of this endemic parasite because they now hold the vast majority of larval stages in this landscape, which is sort of interesting to think about, particularly in an urban environment where you get increased populations of both raccoons and rats and potentially increased interactions between these species, which wouldn't normally occur. And so the rodents definitely host larval stages of the parasite, shown that, but how often are they actually picked up by a scavenging raccoon? Raccoons are not the greatest hunters, but they're thought to be fairly good scavengers. And so the next question is really, how often do raccoons eat rodents? Because it sort of fits in with what we expect out of this parasite life cycle. Actually, the pathology associated with infection is actually thought to be an adaptation on the part of the parasite to increase transmission into raccoons, to make the rodents easier for a raccoon to catch. And so to test whether this trophic transmission is occurring, we set up a bunch of wildlife cameras. So we set up an experiment where we put a parasite-free pet store snake food mouse in front of a camera and then look to see who ate it. And what we found is that almost 90% of these mice were picked up within the 6-day limit that a larval worm stays alive within a rotting mouse carcass. And the majority of these scavenging events looked something like this, which is definitely not a raccoon. But raccoons do consume some mice. So we get our typical scavengers out there, rats, skunks, raccoons, and opossums. And overall, if we break down sort of the amount of scavenging done by each potential scavenger, we see that raccoons get about 16% of the carcasses. So there's definitely a potential trophic link there. They're definitely picking these up. And while they're outcompeted by possums, which is sort of interesting because in possums, they're likely a dead-end host. So anything picked up by a possum is actually eliminated from the system. So definitely a point in the possum's favor. But 16% is certainly high enough to help maintain this parasite in this system. And so there's definitely worms in the raccoons. There's definitely worms in the rodents. We know we're surrounded by this world of wormy raccoons. How worried should we be? We know that this parasite can cause really serious disease in people, particularly in small children. But this is rare enough that when this occurs, this still makes the news generally. But could there be undetected infection out there? What if all these diagnosed cases are sort of the tip of the iceberg? And this is sort of a reasonable question to ask because it turns out that about 10% of us have antibodies to Toxoplasma, which is sort of the cat parasite of crazy cat lady fame, and 14% of us have antibodies to Toxocara, which is a dog nematode that's fairly closely related to Balis ascaris. So what if some of us are also infected with Raccoon Roundworm and we don't know about it? Until fairly recently, this would be a really difficult question to ask. However, there's actually now a way to sample blood samples for this infection and test for antibodies, which is what we did. So we partnered up with the CDC, who's one of the few places that can actually run this blood test. And we collected samples from 150 volunteers here in Santa Barbara County to screen for Raccoon Roundworm antibodies, which provide evidence of current or past infection. And what we ended up finding was that 7% of our totally healthy volunteers tested positive for this parasite, which suggests that those devastating diagnosed cases are really just one end of the spectrum of what Balis ascaris infection looks like. And that this pretty obscure raccoon parasite might infect a lot more of us than previously thought. So what should we do about this? Should we go out and kill all the raccoons? I'd argue no. Or give them all birth control, or eliminate the rodents. Again, probably not likely to be successful. One thing we certainly can do is clean up latrines. I don't think this would work on an ecosystem level, but if there's a latrine in your immediate vicinity, if there's one on your back porch or your roof or your garden, this would certainly be something I would probably clean up. But I'm also going to say it takes a little bit of training to safely clean up a latrine, particularly in areas where you know the raccoons are heavily infected, which should just be an assumption any place there are raccoons. So you can do this on your own. There are resources actually provided by the CDC on how to safely do it, but it's also a service offered by most pest removal companies, and something you can outsource. And sort of, I would argue for people, reducing risk is really about education and latrine removal, knowing what to avoid, and when necessary, actually taking the steps to remove the latrine and remove the risk from your immediate vicinity. Which kind of ultimately boils down to avoiding exposure. And we know that we can avoid exposure. So what about other species? Can other animals out there also avoid risk, this sort of risky sites associated with this parasite? Which sort of takes us on to the next part, the talk, thinking about parasite avoidance. We know that animals can detect and avoid high-risk predation sites. In fact, prey will considerably alter their behavior to avoid predation. And these trade-offs that occur between avoiding predation risks and balancing resource and foraging needs create what's termed this landscape of fear, where prey might choose not to forage in a high-risk area. So we shouldn't, of course, expect hosts to avoid all infectious agents, but when might parasite avoidance occur? I'd argue that it's likely to happen when parasites are worth avoiding, in the sense that it'd be really costly to get infected. This parasite might cause sort of lethal disease. It's got to have high pathology. That's probably worth avoiding. And it has to be avoidable. And to be avoidable, it can't be everywhere. If your risk is the same in all space, there's just no way to avoid that. So you need something where risk is aggregated. And you need those aggregated high-risk sites to also be detectable, right? Because if there's no way to know where the risk is, there's really no way to avoid it. And so in this sense, I'd argue that raccoon latrines, with their high concentration of potentially lethal parasite eggs and a very detectable, obvious cue that they're there, should be a potentially lethal and very much detectable parasite risk. And these sites, if you zoom in on like a single piece of raccoon feces, these are absolutely full of parasite eggs, but they're also full of seeds, undigested seeds from raccoon diets, which means that you might expect animal behavior at these sites to balance risk of disease with foraging preferences, in the sense that an animal may be attracted to these sites because they want to forage on these seeds, or they might avoid these sites because of the risk of disease. And so if you think about how this might balance out, if the pathology is potentially fatal, animal might die from encountering this parasite. It doesn't matter how good the seeds are, you're going to expect these things to avoid these sites. And you see a whole sort of spectrum of different behavior balancing this sort of resource and risk behavior. And so with that sort of as our hypothesis, we went out to test actual animal avoidance and interaction at these latrines, setting up cameras again, this time putting a camera at a latrine and then another one at a matched non-latrine site, and then monitoring animal behavior at these sites. And so we kind of see more in observations of a particular animal at a latrine than at a non-latrine site. We'd mark that as attraction. It's more likely to be seen at the latrine, it's attracted to it. If we see it more often at a non-latrine site, we'd say that's avoidance. And if it's equally distributed at both, it's sort of a neutral, not really interacting with them at all on this sort of spectrum of attraction and avoidance. It's just moving through the habitat. And so we actually go out and measure all this activity at raccoon latrines, representing over 2,000 camera nights and over 2,000 animal visits. We, of course, see quite a few raccoons, a fair number of possums, assorted number of rodents, and the occasional surfer. And overall, about over 30 species came into contact with raccoon feces. And when we start mapping out the activity differences across species, we can start with raccoons, which are sort of a good sanity check. And we look, and looking at the activity difference between latrines and adjacent sites, we see that raccoons are more abundant at latrines. They're showing attraction, which is exactly what we expect. They are the ones making these sites. And we can go through and do this for all the rest of the species in our community. And what we see is that activity differs quite a bit among species, and noticeably the native birds and rodents, where pathology would be quite high, and all seem to show some avoidance of these sites. While rats, of course, don't, although interestingly, these non-native species are also slightly more resistant to this parasite than any of the native ones are. And so what this suggests is that animals can avoid high infection risk sites, which sort of suggests that alongside this predator-induced landscape of fear, there's also a parasite-induced landscape of disgust. And we can kind of depict this like this, which I know is a rather complicated figure, but we'll walk through it. So we think about our landscape out there. There's a variety of different infectious agents, things you can get from other organisms, your fellow conspecifics, things of the same species as you. There's also things you could get from water, things you could get from your predators. So the world is covered in infectious agents, which creates sort of this landscape here of high risk spots where you might want to avoid an area where you're likely to come into contact with those. But there's also predators on that landscape. So animal behavior is going to have to balance risk of predation, risk of parasitism, as well as finding food and mates and living in groups. And so as with predator avoidance, we expect that avoiding parasites will come with costs. For example, reduced social interactions or reduced reproductive opportunities or reduced foraging. And predicting where this avoidance will occur really requires the same trade-off framework we think about for when animals might avoid predators. And we're increasingly recognizing that avoiding predators can have cascading effects on landscapes. And I'd argue that avoiding parasites might as well. It might impact species interaction. If you avoid a parasite, are you then more prone to being captured by a predator? It probably impacts host evolution as hosts evolve ways to avoid parasites, and then in turn, parasites are likely to evolve ways to overcome these avoidance strategies. And a lot of this is sort of theoretical. Do we actually expect to see cascading ecological effects of parasite avoidance? Well, who knows? But I'd argue what we've seen over the last 2 years would strongly suggest that yes, there are cascading effects of parasite and pathogen avoidance that we've all been living through. In the case of COVID, we all are very aware of the effects of infection, but I think most of us are also aware of the effects of infection avoidance. It's altered our social interactions, it's altered our foraging behavior, it's altered our activity. And this has had cascading impacts on other species and the environment. For example, our changing social interactions. How many people got a cold this past 2 years or the flu? We've actually seen huge reductions in the number of other transmissible diseases as we're avoiding interacting with each other. It's changed our foraging behavior in some interesting ways. There's a lot more sourdough. There's also been a lot more backyard poultry, which has actually led to increased outbreaks in salmonella. And then changing in our activity, our driving behavior has led to noticeable changes in pollution levels and things like that. So certainly strong evidence that avoiding infectious agents can have cascading effects in the environment. And so with that, I think I'm probably at about the end of my time. So I'd be happy to take any questions. And I'm also putting up my name and my work email address. So particularly if students have questions about any of this research or research career related things, feel free to shoot me an email.
Okay, thank you, Sarah. Sarah, if you want to pull down your screen, your share screen real quick, then we'll move into the Q&A session. We've got a lot of good questions, and boy, did I learn a lot. So I'm imagining that the audience did as well. I'm going to start out with a first question from Helen Wong, and Helen asks, why raccoon parasites? Why did you choose them to study?
Well, actually, I got pointed in the direction of raccoon parasites, I think by Kevin, who I was really bouncing around between dissertation topics, like walking into his office and being like, I want to dissect a bear. And he's like, why? I'm like, I don't know, I think it'd be cool. And at one point he's like, you should really check out this really interesting raccoon parasite. I was really interested in parasites that might modify host behavior. He's like, people don't know a lot about this one. It's definitely in our local system. And it's thought that the pathology is this severe pathology has evolved to actually increase transmission into raccoons. So it could be a really cool system to study. So, yeah.
Well, it's led you to good places. So, okay, here's the second question. And I'm going to apologize in advance for my mispronunciation of this name. It's Matt Tomaszewski. And Matt asks, do rats have an immune response to the worms that prevents full infection? Or is there a degree of host specificity that prevents rats from being fully infected?
Yeah, so I think part of what's going on with the rats is probably related to an immune response. The rats appear to be very good at walling off the parasites in these granulomas. It particularly, I'm sure at some point they do cause pretty severe pathology, but so as the parasites come in, they come in when the rodent eats the eggs. So these eggs are going to hatch and you have a larval worm that's going to migrate out through the gut and start moving through the tissue. And some places are worse to have a worm than others. And so rats are really good at walling off most of the worms at the gut. So they sort of stop that march of the parasite through. Whereas in mice, a lot more of the worms make it all the way to the brain, where their impacts are going to be a lot more serious. So I think part of that is probably due to an immune response, and also a little bit of a body size thing, that the worms just have farther to travel in the rat. But there actually has not been much work done on that. So it's certainly an area that's worth exploring further.
Great, great. Okay. And here's a question from Scott Cooper. Scott asks, is roundworm infection of rodents and birds always fatal? And does the intensity of infection depend on the species of rodents and birds?
All right. So I would say infection is not always fatal. It is definitely dose dependent. One worm and the animal might be totally fine. Maybe 10 worms, they'd be body size dependent at that point. The smaller you are, the lower a dose is likely to cause problems. But no, certainly not always fatal. And I think, we're actually going to have a hard time surveying the fatal cases out in the wild, because those animals will simply die before we ever get to sample them. And then the second half of the question, oh gosh.
Yeah, he asks, does the intensity of infection depend on the species of rodents and birds?
Yeah, so I would say the intensity, probably the detected number of worms in a mouse, is going to depend on a couple of things. I think it's going to depend on both the contact rate, like how many times that animal comes into contact with the eggs, and then how susceptible that animal is to the eggs. So for every egg that it comes into contact, how many of those actually hatch out into larval worms? And then that sort of determines the number of infections, and then your ability to detect those infections are also going to be a function of how long the animal lives. So if the animal is incredibly susceptible to the pathology, you may never see an infected one because they may immediately die. Whereas if they're incredibly resistant or really good at walling off the worms in the gut wall with relatively lower pathology, like in the rats, they can build up much higher infections. So for the disease nerds out there, it's sort of a function of I'd say the transmission in your basic macroparasite models, actually.
Well, maybe here's another question for the disease nerds out there. This is from Dennis Beebe. And he's asking, would increasing opossum population help to avoid roundworm infection?
I think it could potentially reduce the number of rodent intermediate host carcasses out there. However, it wouldn't change the number of juvenile raccoons that are still getting infected from eggs, because the baby raccoons are just getting exposed to the eggs in the environment. So that would still happen, but it might potentially reduce the number of worms that you're seeing in the older raccoons, which are fairly resistant to the eggs. Also note that possums will sometimes come and just eat feces out of latrines, so they may also be offering somewhat of a latrine cleanup service too, although I'm not sure I'd sort of encourage that necessarily, because the eggs are incredibly resistant to everything. Balis ascaris eggs are kind of insane. They're resistant to like your standard cleaning agents, and ethanol and formalin, and bleach just helps them hatch. They're pretty awful, and they would probably actually survive passage through a possum gut, which would then just disseminate them out into the landscape. So not sure if that would really help the situation.
Okay, so all you possum managers out there, it looks like we're still holding on using that as a tool. All right, so here is here's another question here's a question from Jesus Munoz, and Jesus asked, was this a form, and this meaning your research, Sarah, was this a form of applied or basic science? He's saying it seems like both to me. And would you mind sharing a bit of the purpose or implications of this study?
Yeah, I'd say it's both. And I think a lot of research, particularly disease-related research, kind of crosses both applied and basic. And I'd say, understanding what the distribution of worms are, and that young animals young raccoons have more worms than older ones could potentially impact how we control this parasite. For example, it's often suggested that we should just cull the raccoons. If we just remove raccoons, we keep trapping them and taking them out, that would be a great way to reduce the parasite. But often when you try to cull an animal, you can't catch them all. You can never catch them all. And that can actually change. So the animals that are left might actually have more babies in response. So by culling, you may actually be shifting the whole age distribution down into the most heavily infected animals. And so by knowing that the juvenile is the most infected, it would maybe suggest that culling is probably not the best way to manage this parasite. You probably actually want to keep your oldest raccoons and maybe explore options like antihelminthic baits, where you just distribute marshmallows full of dewormers out into your landscape and keep the raccoons and just keep them worm free. So I think there are some certainly applied aspects thinking about parasite control, particularly for a parasite like this, where you very much do want to understand how to control it.
Okay, thank you. And Jesus also says, great presentation for Halloween too. So maybe this will inspire some ideas out there. Okay, so we have maybe a few more minutes left for a couple questions, and they keep coming in. So I'll try to get to as many as I can. And everybody, thank you for your attendance and attention. This is a question from Diane Galvin. And Diane Galvin asks, is there much data on dogs, like pet dogs, and level of raccoon roundworm infection? How are those species related?
Yeah, that's actually a great question and something I kind of just completely skipped over. So raccoon roundworm is almost always in raccoons, but it can also use a couple of other species as a definitive host. So things that are pretty closely related to raccoons, the worms kind of get in there and like, I'll make this work. So skunks and dogs actually fit into this sort of dog-like carnivore group. And so you can actually get mature raccoon roundworm infections in dogs. I don't think we know how common this is because just looking at these worms, they actually look quite a bit like Toxocara, this dog roundworm that's incredibly common. And particularly the eggs look pretty similar. So vets doing parasite diagnostics would generally probably just call it Toxocara or roundworm and then treat the dog and eliminate the infection. So it's good that your standard anti-parasite medications for dogs are quite effective on this parasite. However, I think it's certainly something to be aware of if you have a dog that likes to eat wild animal poop. It is probably being exposed to wild animal parasites, and Balis ascaris is one that you really don't want your dog to get. There are cases where the larval worms have stayed larval and cause pretty serious disease in young dogs. And in an older dog, while the dog may not experience much disease from having a couple of big worms in its gut, dogs are what we term indiscriminate defecators. Unlike raccoons, dogs don't form a latrine. They just poop wherever they feel like it, which means that potentially if they're, have some of these worms in there, they're putting these parasite eggs out all over the place, making them much harder to avoid and clean up.
Great. Okay. And so here is our last question. And we have just a minute left for it. So maybe you can give us just a little bit of a teaser, Sarah, maybe point us to some other resources if possible. But this last question is from Scott Cooper. And Scott says, could you tell us about the poisonous African rats?
Yeah. So for part of my postdoc research, I started studying the African crested rat, Lophiomes imhousei, which are these giant black and white colored rats. They're aposematically colored, so they have warning coloration because they actually coat themselves in plant toxins from the poison arrow plant, which is the same plant that's been traditionally used to make arrow poisons in sub-Saharan Africa. And these rats actually chew on this plant and then they lick it into their fur. And when they're annoyed or afraid, they actually raise this fur crest, exposing these wild poison stripes on the side. And if a predator comes and tries to get a mouthful of the rat, it just ends up with a mouthful of these poison covered hairs. And it's a cardinalide, so closely related compound to the stuff that's in monarch butterflies, but also the human heart medication digoxin. And so these are really potent sodium pump inhibitors. So they actually bind to the sodium potassium pump that's present in essentially all animal cells, which is what makes cardiac glycosides or cardenolides, effective drugs to be used to treat conditions like congestive heart failure, but at the same time, really dangerous drugs because you overdose just a little bit and you essentially stop all the muscle contractions and rapid poisoning. So I've been studying this weird African rat for the last couple of years, mostly in Kenya, which is probably the easiest place, or I'd say the safest place to work on them. And we recently had a paper come out on their social behavior and are currently working on some stuff more on their chemical ecology and their microbiome right now. But a very cool system, but a little farther from home.
Well, wonderful. And if we start seeing those African rats at Coal Point Reserve, I'm going to be really worried, and I'll know who to call, Sarah. So I just wanted to thank you, Sarah, for your presentation. Thank you, Cris, also for your presentation. Next week, please, everyone, come back and join us again. We're going to head over to the mountains with our host of the Valentine Eastern Sierra Reserves. And we'll hear from Dr. Joe Cutler, who's going to present From Fishes to Frogs, Aquatic Ecosystem Restoration in Yosemite National Park. So don't miss out. And just a reminder that tonight's presentation will be posted on the UC Santa Barbara Natural Reserve System YouTube page tomorrow. So please, check that out if you'd like to catch any of what you saw tonight again. And thank you, everybody, and have a great night.